are merging telemedicine and nanotechnology. I just want to remind you all that the bus is leaving for the hotel, and we're meant to be downstairs near the reception area at 10 minutes after 7, as Temple's just told me. Our first speaker in the emerging telemedicine and nanotechnology is Christopher Phoenix, who received a bachelor's degree in symbolic systems and a master's of science in computer science from Stanford University. He's been actively involved with nanotechnology since taking Eric Drexler's class in 1988. And in 2002, he co-founded the Center for Responsible Nanotechnology, where he served as the Director of Research from 2003 to 2007. He's also a published author in the fields of nanotechnology and nanomedicine. Mr. Phoenix, please. That advances the slides. You got it.
Okay. Well, I'd like to thank you all for listening at the end of a long 2 days. I'd like to thank Dr. Rothblatt for inviting me. This talk will be different from almost all of the talks here because I have no laboratory results to present. There have been some amazingly inspiring talks here, and I hope that this talk will be inspiring in a different way. I will be talking about things that we can't build yet. Why are they interesting? Right now, we can build things like this, little teeny nanofibers, much smaller than a human hair, with all sorts of interesting mechanical properties. We can simulate things like this, and even this, which is molecular machine components, gears and such, and we can tell that in simulation, in high-quality simulation, they have excellent performance characteristics. Extremely low friction, extremely high power density, effectively zero wear. Basically, once you can build things like this out of atoms, one atom at a time, you have a real sweet spot in machine performance. Now, building things one atom at a time takes a very long time. At least for a tabletop scanning probe microscope, it would take about 10 to the 18th years for it to process its own mass in atoms. The good news is that that time scales as the fourth power of the size. So if you brought it down to a micron, if you could build a scanning probe microscope about a micron big, it would take about a month to process its own weight in atoms. Bring it down one more order of magnitude to 100 nanometers, and it could do it in minutes. At that point, you can start to think about building a scanning probe microscope that can build another of itself, obviously under computer control, in a time short enough that you can start to talk about nanofactories, about once you get all the way down to the bottom, building back up to vast arrays of these things, as explained by Dr. Merkel, talking about the chemical reactions it would take, and Timur Tothfail talking about some of the things we might build along the way. So this talk is going to start from the assumption that sooner or later we will reach this sweet spot where we can have nanomachines building nanomachines and exponential manufacturing, building as many of these things as we want. So this is my history. One thing I want to mention is my first publication in nanotechnology is the vasculoid concept, co-authored with Robert Freitas. And that's the idea of replacing the human blood volume with a branching robot that fits inside the blood vessels. Now this was fun, it was highly exploratory. It turned out to be dozens of pages long to even touch on the architecture that was needed to pull this off, to do all of the necessary functions of blood in a non-fluid robot. In 2002, seeing that not enough people were talking about even the possibility of molecular manufacturing, I co-founded the Center for Responsible Nanotechnology, spent the next 5 years basically working to get people aware that this stuff was physically possible and sooner or later it was going to happen, and the power of the devices would be world-changing. In this talk, I'm going to talk about a very, very small area of medicine, which is small internal devices. I'm going to talk about a very small area of nanotechnology, which is molecular manufacturing. And I'm going to talk about a very small area of communications, which is fairly short-range communications, low-power communications, to fit the requirements of small in-body robots. So there's vast reaches of stuff that I will not touch on. Okay, what is a robot? It's a machine with programmable behavior. Basically, anything that we can build and then decide what we want it to do is what I'm considering a robot. So if we design a medical device that only does one thing, you inject it and you let it do its thing, that's not a robot. That you design in advance. You don't have to control it once it's in the body. But if we build a platform that can do any of several things, maybe diagnose a condition, report out, and then take instruction for what to do about it, that's a robot. And this does not imply a high degree of autonomy. It does imply a degree of signal processing and local decision making. And basically, robots require either a lot more internal smarts than I want to speculate on, or they require communication. Okay. Today's molecular technologies are on the order of single molecule sensors, energy transducers, rafts of molecular devices that do several different things at once. These are very cool. What I'm talking about is something considerably more complex than that. Here is, just to level set, here is a device of the kind that might be inside one of these robots. This is, for example, blood plasma. This is a highly purified reservoir of one particular molecule that's in the blood plasma, for example, oxygen. And if you look at the scale, this whole disk is about 10 nanometers wide. That's about the size of the lines that we can draw today with the best lithography techniques. On the other hand, the features of this are only about 1 nanometer wide. That's one order of magnitude. That's not inconceivable. And that's a few atoms wide, 1 nanometer. So we can start to see how something like this might be built. And devices of approximately this complexity have been simulated and more or less work. I mentioned a 100 nanometer scanning probe microscope. You could fit many thousand of these into that volume. So 100 nanometers is a constraint, but it's not an extreme constraint for fitting lots of devices into a nanomedical robot. The actual robot would be about 1,000 times that volume, about a cubic micron. That seems to be a reasonable size, big enough to swim, small enough to fit into a cell, and small enough to build a lot of them. You could have maybe one nanobot per 1,000 cells if you build them a cubic micron, and if you're not limited in the number you can build. Okay, so I talked about high performance, and I implied large quantities. Basically, if you can build scanning probe microscopes with scanning probe microscopes, then you can build as many of them as you want. It doesn't take very many doublings to get from an atom to a kilogram, about 60. And when you can build with atoms, you can engineer a vast array of molecular machines to do things like sorting, sensing, computing, power generation, signal generation, basically everything you would want in a medical robot. Something I will not have time to talk much about now is the broader implications of these products, but let's just say it doesn't take many more doublings to go from a kilogram to a gigagram, and planet scale engineering looks like it goes from decades to months. Okay, this comes from an animation about 4 minutes long that I will not have time to show of how a nanofactory might be architected. And I've written an 80-page paper on nanofactory architecture, published at the Journal of Evolution and Technology. which basically says, yes, you can make a kilogram scale product that contains 10 nanometer scale machines that can build about a kilogram per hour in a kilogram scale factory. You can deal with radiation damage, you can deal with power supply and heat, feedstock supply. Everything I could think of you can do in a tabletop factory that can build another factory in a few hours. Or can build thousands of therapeutic doses of nanomachines in an hour. Okay, medical problems to solve. I'll just briefly mention these. Biocompatibility, we've heard a lot about. Power supply, you don't want to use more than 10 to 100 watts total in the human body. If you have 10 to the 11th nanomachines, then you've got on the order of 10 to 100 picowatts per nanomachine. This is also about the limit of what you can get from body glucose and oxygen. So figure a power budget of 10 to 100 picowatts per cubic micron nanomachine. This is going to limit the communications that you can do. Heat dissipation is not so much of a problem. It's certainly not a problem for single nanobots. In large aggregate, it is a limitation. You don't want to cook yourself. You don't even want to break a sweat. Preferably, you'd rather not know the nanobots are there, except you just feel better. Sensing, there's all sorts of things to sense, everything from blood pressure to molecular concentration to neural impulses. And sensing is related to communication, so I'll talk a little bit about it, but the main focus of this talk is communication, how to get information between the nanobots, to the nanobots, and from the nanobots. Okay, so today we have things like hormone pumps, pacemakers. We have some molecules that are going into the body, and some of those molecules are performing fairly intricate functions. Anti-cancer packages, liposomes, anti-cancer packages that include liposomes, and some of these are triggered externally. So we're starting to see already a need for communicating to internal devices. However, the kind of devices I'm talking about, you'll have plenty of room for internal logic. You won't have to communicate by heating them up. You'll be able to communicate by sending in an RF signal at a megahertz. It'll be able to read the signal, process it, and basically you'll be able to get, 100 kilobits a second or more of data coming in and do something sensible with that data. So the devices that I'm talking about will have onboard computers. If they'll exist at all, onboard computers are not much more difficult. So, a few cubic microns, as I said. Molecular intervention, meaning they'll be able to manipulate individual molecules as desired. For example, to crawl into a cell and stitch together the coat, the various layers on the outside of the cell. They'll be able to intervene functionally. A robot of this size can detect and trigger nerve impulses, can release biologically significant amounts of chemicals, at least significant within a single cell. And in aggregate, of course, these devices could release enough of a particular chemical to affect the whole body. So there are various ways that these things can affect body function in addition to molecular manipulation. If you put in this many robots into the body, and there's really no reason not to, because you can build them small enough, you can fit an awful lot of cubic nanometers into a cubic micron, a billion in fact. You can put them about every 100 microns, so you'd have maybe 1,000 cells per nanobot. At that point, you can start to think about keeping track of what every cell in the body is doing. Not in real time, but you could visit each cell in the body every day or maybe every hour if you wanted to. This is obviously going to generate far more data than the bandwidth you have, so compression, onboard analysis, these are going to be very important, and I don't have time to talk more about them. Okay. I'm also not going to be able to talk much about why medicine is so hard. You have multi-level systems. You have constant influences from the environment, from other parts of the body, at each of these levels. You have pathogens that you don't know they're there, maybe until they start causing problems. Some pathogens can dress themselves up like body cells, and it's not obvious that it'll be easy to detect that any more than our immune system can detect it. You have pervasive degeneration, where things in the body just sort of fade out over time, and repairing this will be somewhere between difficult and extremely difficult. I do not say impossible, but extremely difficult to deal with accumulated DNA damage, for example. And it might be a full-time job for these nanobots to go around and unlink the cross-linked proteins and break apart the plaques. And each of these functions, of course, will have to be separately researched, designed, and probably controlled from outside to some extent. Disease identification, obviously, if you're not feeling well today, okay, I have a headache. Well, what do I tell my little 1-micron nanobots to look for? They can't look for a headache. They may be able to look for neural impulses. They may be able to correlate over time that headache with biomarkers. But there's vastly more problems to solve than just communication. But I have 20 minutes. 5 minutes now. Okay. So you want to learn the medical status. You want to send that between the nanobots and send it out of the body for use by the user and the doctor. You want to be able to control the robot behavior, and you want to be able to provide some infrastructure to the robots. For example, it helps if they know where they are, and it helps if they can act jointly. If you wanted to, for example, present an image to the optic nerve, they'd have to fire in tightly coordinated sequence and spatial location. Okay, just a few examples of some sensory capabilities. A cubic micron detector could detect about 10 million molecule types. That's about how many receptors you can fit in. You can detect displacement, motion, force, pressure, sound, temperature. You can detect electric fields. You can detect cell structure. And all of this is online in Nanomedicine Chapter 4. The whole book's online, and it's definitely worth reading. It contains theoretical limits. It contains implementations that get fairly close to those theoretical limits. So if you want to think about where your devices can go, this is a great book to read. Just for example of some trade-offs, if you only need to read temperature within about 31 millikelvins, then you can do that every nanosecond in a 57 cubic nanometer sensor. If, however, you want a microkelvin, you need about a cubic micron sensor and 100 microseconds to read it. So a lot of sensory information, the trade-off is with size, energy, and time to get the level of precision that you need. Now, some things you can't do without getting far too big for a free-traveling robot. You can't make an electromagnetic antenna that works. You can, however, receive electromagnetic signals. Another thing you can do is you can detect a single proton mass difference. You can basically weigh an isotope in about 10,000 cubic nanometers and do about 100,000 of those a second. You can detect displacements of 10 picometers and forces of 10 piconewtons fairly easily, in fairly small sensors. What this boils down to is acoustic communication looks like the best way for one robot to communicate with another. Electromagnetic, as I said, you can receive from outside the body at hundreds of kilobits or maybe megabits per second, but you cannot transmit on electromagnetic wavelengths with very small robots. Acoustic, you can send the necessary distance, the 100 microns or so. Chemical signaling looks quite inefficient for reception, but you can transmit at fairly high bandwidths as long as the chemical is going to be excreted and processed outside the body. There will, of course, be delays on that. But if you want to record something and process it later, As long as you can build a molecule that'll be filtered out in the urine or dialyzed out of the blood, and as long as you can get all of the molecules that are produced and analyze them individually, which is not something you can do in 1 cubic micron, but it's something you could easily do in a desktop appliance, then you can get quite a lot of information out of the body that way. It takes about 60,000 picowatts to transmit continuously at 100 megahertz, which appears to be the most efficient frequency, acoustic frequency at these distances. Longer distances just get less efficient overall. And you don't need to go much beyond 100 microns. If you have robots circulating through the capillaries, they get that close to pretty much everywhere in the body with some obvious exceptions. Now, if you need to limit yourself to a few picowatts, you can get a few kilobits per second, which is probably adequate for a whole lot of control and reporting tasks. This is a few kilobits per second per nanobot, and remember you've got 10 to the 11th of them in the body. So this is 10 to the 15th or 10 to the 16th bits per second in aggregate. This is far more than any system can make sense of today. So it's enough for any technology that we can start to think about designing today. It will not be enough by the time we get around to it. We'll want to do more things and we'll know how, but for things that we know we will want to do, this is enough. Radio, I already said this. Incoming only, you can send in a lot of data into the body. So once you have reported out the condition, you can program your robots almost individually. It would take maybe a day to send in information to each cubic micron of the body. I calculated that. Never mind. But to program the robots individually with this channel is probably not quite possible, but you can program them in very small aggregates with incoming signals. And okay, bigger questions that are raised by this level of medical technology. It seems fairly clear that you'll be able to not only correct diseases, but you'll be able to enhance body performance. One example has been given already, which is you inject a therapeutic dose of respiricides, and now you don't have to breathe for the next few hours. This usually will not cure a medical condition, but there will be a whole lot of extreme sports people who want to use it. Now, as a doctor, are you going to prescribe this for this purpose? And as a doctor, are you going to fight against it being available through non-doctor channels? Because people will want it and they will try to get it one way or another. Question about interactions between the patient and the robots, both in the matter of patient choice and in the matter of how the interaction is implemented. Neural stimulation raises all sorts of issues. Issues of what happens if you have a bug, meaning a software bug, and what happens if it turns out that stimulating certain neural channels is subtly a bad idea, if it tends to rewire the brain. Also, what about involuntary stimulation? And obviously, any technology this powerful will have lots of destructive uses. And you can certainly get enough information both into and out of the body with even very small robots that might be put in there purely for therapeutic purposes. You just stick in a temperature sensor, an electric field sensor, a sound sensor, a chemical sensor, and now you can tell what every cell had for breakfast. This is considerably more information than you can get from a person's genome, and already that's raising all sorts of ethical questions. So when you can tell what a person's body is doing at the level of their individual cells, in a sense, you know more about them than they do themselves. I saw a calculation once that it should be possible to outmessage every neural firing, and that raises very interesting questions about mental privacy. Okay, you can email me with questions. The nanomedicine book, the table of contents is here. As I said, the entire book is online. And I believe the second volume, Biocompatibility, is now also online and should be pointed to from this page. And the organization I mentioned that I co-founded is crnano.org. And we have a lot of information about molecular manufacturing and the broader implications there. Okay, thank you.